Well, good morning, good afternoon, or good evening, ladies and gentlemen. Welcome to another installment of our video PowerPoint series for Unit 6, DNA, RNA, and Protein Synthesis. Today, what we're going to be talking about is primarily RNA and how RNA is used to go ahead and create proteins via protein synthesis. So, without further ado, let's go ahead and get this thing started. I will see you on the next slide. So here's the main plan, alright? The big question is DNA controls everything inside the cell, right? But how can DNA do this if it can't leave the nucleus? What happens is we go ahead and send a messenger, in this case, messenger RNA, or mRNA for short. Now remember, DNA contains the code for making protein. DNA can't leave the nucleus, so what it does is it has RNA to help out. And the RNA is what actually makes the proteins through the use of the ribosomes. So the first thing we need to talk about is ribonucleic acid, or RNA. RNA is a molecule that controls the production of proteins for cells. A strand of RNA is made of repeating units or monomers called nucleotides, just like in DNA. And if you remember what makes up a nucleotide, we have three components. We have a phosphate, a 5-carbon sugar, which in this case is going to be ribose. But in this case, the nitrogen base is always going to be just a slightly different. As you can see, we have A, U, C, and G, which is adenine, uracil, cytosine, and guanine. So RNA versus DNA. There are three major differences between the two. RNA being single-stranded, using ribose instead of deoxyribose, and using uracil instead of thymine. So as you can see from the picture right down here, the RNA molecule is made of phosphates and sugars, just like we have right here. So we still have the same backbone, but the differences are, instead of using thymine, designated as T, we use uracil, designated as U. So let's go ahead and look at the main differences between DNA and RNA in a little bit more depth. So DNA is a double strand, whereas RNA is a single strand. DNA uses the sugar deoxyribose, whereas RNA uses ribose. DNA contains thymine, where RNA contains uracil. And DNA has to stay inside the nucleus, whereas the RNA can leave and pretty much float freely wherever it needs to go. So it can go from the nucleus to the cytoplasm, ultimately hooking up with the organelle that makes our protein, the ribosome. So we have three types of RNA. We have messenger RNA, known as mRNA. This is a single uncoiled strand, and it serves as a pattern for the assembly of amino acids. Now, if you think back to the biochem unit, we know that amino acids are the monomers of our protein. We have tRNA, or transfer RNA. These carry amino acids to the ribosome. And these are also single-stranded. So the transport or transfer RNA goes ahead and brings the amino acids to the ribosome to be assembled into our proteins. We also have ribosomal, or rRNA. It's a globular form that primarily makes up the structure of the ribosome itself. So we have three types: mRNA, the messenger, tRNA, the transfer, and rRNA, the ribosomal. So the first process we're going to talk about in protein synthesis is called transcription. It's the process of making mRNA from a single strand of DNA. And if you remember DNA replication, there should be a lot of bells that are ringing here. So the nitrogen bases in RNA always pair up to the complement on the DNA strand. So cytosine is always going to go with guanine and vice versa. And in this case, adenine is not going to go to thymine, but it's going to go to something called uracil. So adenine binds to uracil, guanine binds to cytosine. And just for the record, if there's any thymine on the DNA strand, that is going to pair up with adenine, just like in DNA replication. So the steps in transcription. Step number 1. The enzyme RNA polymerase unzips the complementary strands of DNA into 2 strands. Then we have the RNA nucleotides that bond to the single strand of DNA. And then the finished mRNA is released and the 2 DNA strands rezip. So if you go ahead and bring your attention down to the bottom here, you'll see how this all works. So we have our RNA polymerase that's going through and unzipping our DNA molecule. Once we have the 2 strands unzipped, as you can see right here, we have matching bases being paired up. So we're getting a copy of one of the other strands. Except this time, instead of using thymine, we have uracil instead. So the RNA polymerase goes ahead, unzips, makes a copy of the strand, and rezips as it goes down the DNA. This continues until it hits a stop point. So once we have a completed copy of the DNA strand, they go ahead, zip it all up, and the RNA then leaves the nucleus. So, if you want to go ahead and take a little bit more of an in-depth look at this, it's a very good animation. Right down here below, if you go ahead and click on the link, you should be able to go to this transcription animation that shows you exactly what we were just talking about, but in a very nice animated form. So if you want to go ahead, check it out. I'm just recording, so I'll see you as soon as you come back. So, mRNA processing. We have enzymes here, very specific enzymes that remove or cut out something called introns, because they interrupt the coding sequence. What they're doing is they're in the way. We don't want them, so we actually get a chance to cut them out. So introns interrupt. We take the introns out. We don't want them. Whereas exons are left behind to be expressed or translated as needed proteins. So in this case, what we do want, we want the AUG, but for our next line, for our next codon, we want CAU. Then for our third codon, AGC, and our fourth, CUA. So in this case, we don't want this U, or this uracil, nor do we want these 2 Gs. So we actually have the opportunity to go ahead and cut them out. So this way we get the codons we want, and not the ones that we don't. So again, like we said before, if you go ahead and take a look at this, we have exon, intron, exon, intron, exon. So basically what that means is, when we go ahead and transcribe these, going from the DNA to RNA, we can actually go ahead and remove certain bits. So here, we went ahead, snipped out the introns, and all we have left are the pieces of the code that we wanted. So, we have a checkpoint here. I'd like you to go ahead and transcribe the DNA strand into RNA. So we have TAC, TCG, TCC, ATA, GGC, and ATC. So, why don't you go ahead and pause the video, or not, it's your call. While I'm talking, you might have already figured this out. So I'd like you to go through and make the complementary base pairs to take this from a DNA strand into an RNA strand. Remember, T on the DNA is going to go to A on RNA, A is going to go to U, C is going to go to G, and G is going to go to C. So go ahead. Why don't you go ahead and transcribe it, and then in a little bit here, once I'm finishing, up my tea, I'll show you the answers. That is fantastic. So what we should have is AUG, AGC, UGG, UAU, CCG, and then finishing it up with UAG. Just like we said before, A goes to U, T goes to A, G goes to C, and C goes to G. So protein synthesis. The bases in the mRNA code for which the amino acids will make a functioning protein. A group of 3 sequential bases on an mRNA strand is what we call a codon. Consider it like going ahead and using letters to create words. And then you go ahead and put the words together to make a sentence, right? So depending on which combinations of letters you use, that's going to determine what words you make, and ultimately what sentence you form. So, if you look at this, our first codon would be GCU. 3 letters. Our second, ACG. Our third, GAG, and so on and so forth. Now these codons are going to code for specific amino acids that we're eventually going to use to go ahead and create our proteins. So there are a possible 64 codons that code for about 20 amino acids and a start-stop signal. The genetic code is universal among all organisms. So, that's right, there are 20 amino acids in bananas, 20 potential amino acids in chimpanzees, and also chickens. So depending on which way the codons are arranged and which proteins are created, that ultimately determines, well, what you are. A banana, a chimp, or a peep-peep. Yes, I said peep-peep. Deal with it. So the genetic code. By going ahead and using this, what we call a codon wheel, we can determine what bases and what codons will code for what amino acids. So the first thing you should do is go ahead and separate your bases into your codons. Remember, a codon is how many bases? That would be 3. So we go ahead and divide up our bases into our codons. So then we have to figure out what codon is going to code for what amino acid. Now the way this works is the first letter is your inner ring, the second is your second ring, and the third is your final ring. So if we start off, let's go ahead and take a look here. We have GCA. So we start off with G. So we know we're going to be in this area right here. So we have GCA. The next letter, so our second ring, is C. So now we go from our second rung to our final rung and find A. So GCA would code for alanine. How about ACG? So if we go ahead and take a look at this one, we start off with A, we move to C, and then we move to G, which would code for threonine. UUG, we start off with the U, go to our second U, and then we go to G. So UUG would code for leucine. So I'm going to give you a little bit of time here. I'd like you to go ahead and code for the last 2 codons. Like I said, pause the video if you need it. Okay, pause. Okay, did you do it? I'm going to assume you did. So what we have next, CUA would code for leucine as well, and CUG would also code for leucine. So the steps in translation. So once we have the DNA to RNA through transcription, we have to go through translation, which is RNA to protein. So with translation, the process of using RNA to assemble amino acids into a protein, the first step we need to do is the mRNA moves out of the nucleus and attaches itself to the ribosome. Now if you remember, the ribosome is where the protein is created. Step number 2 is the bases are read off 3 at a time, so 1 codon at a time. The tRNA transports the correct amino acid to the ribosome. So as you can see here, the mRNA goes ahead and works its way into the ribosome. Once it goes ahead into the ribosome, it's pulled through and read off 3 bases at a time. While it's being read by the ribosome, it's calling transfer RNA to go ahead and bring the corresponding amino acid. So if you look here, on our codon we had AUG, so the transfer RNA brought the correct base pairing, UAC, along with methionine, the correct amino acid. So the anticodon on the tRNA bonds to the complementary codon on the mRNA. What this means is, for the amino acid to actually be let go by the tRNA, it needs to create a bond with the mRNA just for a second. So when it does this, this was the 3 complementary bases to AUG, which was UAC. The amino acids then form peptide bonds to form something called a polypeptide. The polypeptide is what is eventually going to become our protein. So the stop codon on mRNA ends the process and the new protein is released. So the stop codon goes ahead and tells the ribosome, hey, we've got what we need, you can stop. We appreciate it, but we have what we need now. Thank you for your service. So, like we said before, we've got, you can't really see that. Let's go with this. So we have our start codon, AUG. The tRNA brings the anticodon, UAC, as well as the first amino acid. Once it bonds, it lets go of the amino acid, and puts it inside the ribosome. Now, as the mRNA is read, as it's being pulled through, more amino acids are brought, and they're being strung together via peptide bonds to create what we have here, a polypeptide. This is going to continue until we eventually get to the end codon, or the stop codon, where the last amino acid will be strung together via the polypeptide, and ultimately released. So, it's your turn. Go ahead and translate your codons into amino acids. Now, just like we did before, if you look inside your workbooks, I believe you'll find a codon wheel. Use the codon wheel to go ahead and figure out what amino acids you'll get from your codons. Your codon AUG, CAU, AGC, and CUA. Remember, the first letter is your inner ring. The second letter is your second ring, and the third letter is your final ring, which tells you which amino acid you'll get. So, go ahead, hop to it. I'll be here. I'm not going anywhere, I promise you. By Jove, that tea is wonderful. So, here we go. AUG codes for methionine. CAU, histidine. AGC, serine. And CUA, leucine. So if you'd like to go ahead and take a look, there's another video down here below that shows you protein synthesis. If you want to go ahead and give it a look, it might simplify what we just talked about. Well, ladies and gentlemen, that's just about going to go ahead and finish things up for us today. I'd like to thank you for joining us as we discussed RNA and protein synthesis. So in conclusion, what we talked about today was RNA, which stands for ribonucleic acid. The differences between RNA and DNA are RNA is a single strand, it contains ribose instead of deoxyribose, and it also uses uracil instead of thymine. Now, what we also talked about was protein synthesis, how protein is created from basically DNA to the end resulting protein. The first step is when we go ahead and create our RNA strand from DNA. We call this transcription. So with transcription, we go from DNA to our mRNA copy. The next step, translation, involves mRNA, tRNA, and eventually gives us our protein. So translation, what we get is we go ahead, start off with the mRNA, and eventually get our polypeptide or our protein. Well, that's about going to go ahead and do it for today. I'd like to thank you all again for joining us, and until next time, I will see you in the next video. You all keep it classy.